there is an alternate state that is always being triggered by starving. And so what do if you have nothing? Well, what do you do if you're a bacteria? What do you do if you're a mouse? And what do you do if you're a primate? Clearly, some of the same pathways, they seem to come up over and over.
Aging. Like gravity, it pulls on each of us. Why do some of us age without illness? How do our bodies and minds experience aging at the cellular and molecular level? What's the future of aging in our society? And maybe, most importantly, what can we do about it today? My name is Gordon Lithgow, and here at the Buck Institute in California, my colleagues and I are searching for and actually finding answers to these questions and many more. On this podcast, we discuss and discover the future of aging with some of the brightest scientific stars on the planet. We're not getting any younger, yet. Hello everyone, welcome to the podcast. Today's going to be great fun because I get to talk to my friend and colleague, Valter Longo. Valter's at the University of Southern California and has been running a research program that really gets into the way that aging is affected by diet, and specifically how low-calorie diets can affect all sorts of age-specific diseases, including cancer. I think we're going to learn a lot from this one. Valter, thanks for taking the time to do this today. How are you?
Good. How are you?
I'm good. So you're in Europe right now?
I'm in Southern Europe, in the land of centenarians. Yeah.
You know, we were recently talking with Sachin Panda, and there's all sorts of things we talked about: caloric restriction, intermittent fasting, fasting mimicking diets. So, Valter, could you take a brief amount of time and just define some of the terms that are floating around? Caloric restriction, I think most of our listeners have a reasonable knowledge of that, but what's that compared to fasting? What's that compared to starving? And what's that compared to intermittent feeding?
Yes, so fasting is just not eating at all. In most cases, anywhere from 12 hours to 20 days, right? And time-restricted eating refers instead about the time you restrict your eating to, right? So meaning that if you start eating at 8 a.m. and you end at 8 p.m., that's 12 hours, right? 12 hours of eating, and then at night to 12 hours of fasting. And so that's time-restricted eating. And usually the 12-hour fasting is the minimum, and then it goes all the way to, let's say, 20 hours of fasting. So some people, or even 22, some people say fast for 22 hours and eat for 2 hours, right? Then fasting mimicking diet instead is something that we developed many years ago because we started the first cancer trial at USC Norris Cancer Center. And we thought everybody's going to do water-only fasting. It was water-only fasting for 3 days, 2 days before chemo, 1 day after. And we realized, and the oncologists realized, and most people did not want to do water-only fasting. It's really tough to do. And then we understood why people should do it in a specialized clinic. And then, the National Cancer Institute and the National Institute on Aging then funded research together with others to develop a fasting mimicking diet, right? So can you let the cancer patient eat and achieve lots of the changes that you will normally see with water-only fasting? So we had lots of markers that we looked for, like IGF-1, insulin, ketone bodies, glucose, IGF-PP1, etc. Right? So then that's what a fasting mimicking diet is. Now, just to summarize. 4 days for cancer patient of a fasting mimicking diet, about 5 days for normal subjects and diabetic subjects, and then it goes to 7 days for multiple sclerosis, autoimmunities. In the trials, at least that's what we're testing, right? So 4 or 5 or 7 days based on with different compositions.
I guess the question really is, right now, what do you think's the champion?
Yes. So I think that probably if you think about safety as well as efficacy and proven efficacy, for the time restricted eating, as such knows, I like more the 12 hours, 12, 13 hours of fasting versus the 16 hours. 16 hours, most people skip breakfast, and that's associated with lots of problems. And the gallstone formation, so the 16 hours has got a lot of bad associations. Now you could argue that that's something else. It's not really the 16 hours that is causing that, but it's not a good start. I think it's not good to start with shortened lifespan, higher mortality, more cardiovascular disease for those that skip breakfast, right? And in most cases do this very fast, long fast daily. But the 12, 13 hours, I think that there's lots of positive data, and I think it's also very safe. I always say I've never seen a negative study about fasting for 12, 13 hours. Of course, it's not as effective as the 16 hours, but I mean, all considered, it's probably a very good way to go. And then, of course, I'm a big biased fan of periodic fasting and particularly fasting mimicking diet. And so the idea would be that get away in a sense from things that you have to do every day for those that cannot do things every day, and that's the great majority of the population, and say, well, could you do this 3 times for 5 days a year, right? So 15 days a year. And I mean, of course, does it work or not? Well, we'll have to see. But lots of the trials that we completed indicate that it does work.
Let's go back a little bit into the science and just how the different diet regimes that we've talked about interact with what's known from worms and other aging systems. What's the connections between exposure to nutrients, the time that nutrients are taken in, and the rest of the aging biology that we think we understand?
Yeah, I think it's very, very much conserved, and maybe more conserved than we realized. That whether you're a bacteria or you're a person, there is an alternate state that is always being triggered by starving, and not necessarily calorie restriction, right? So maybe calorie restriction was like an in-between, like coincidental thing, right? Somebody 100 years ago, McKay and company, ended up, you couldn't starve a mouse or a rat for years, right? So they calorie restricted them. And I don't know if it was voluntary or not, but maybe the origin of all of this has always been starvation. And so what do you do if you have nothing? Well, what do you do if you're a bacteria? What do you do if you're a mouse? And what do you do if you're a primate? And so I think that clearly some of the same pathways, Taurus kinase and some of the growth factors and lots of other genes that have to do with growth, they seem to come up over and over in all these different systems. And so I think that's probably a fundamental property of all organisms.
And you've shown in your reviews the interconnectedness between different aging systems, but also, do you have a particular interest in fat? You seem to be publishing in that area quite a bit.
Not really, but I think I'm very interested in starvation responses, and the fat is, of course, part of the survival program under starvation response, right? So storing fat and then burning it, right? And so in yeast, of course, we saw back in the days the trehalose and the glycogen being accumulated, and then as you move to worms and higher eukaryotes, then you see more of the fat storage process occurring in some of these mutants. It seems to be one of the conserved behaviors, right, or mechanisms that all these organisms entered when they were starved, or when they were mutated in these starvation response pathways. So, and then of course now, because 50% of Europeans and 72% of Americans are overweight and obese, it's a very important area, to accompany the anti-aging effects, right? So certainly this understanding of the fat pathways. I mean, the idea, of course, is can you deplete fat or reduce fat and without affecting lean body mass, right? And without affecting frailty, right? So, I mean, this is really something that we're focusing on. Can you get a mouse to live long with low fat and strong, right? And it's not so easy, right? Based on just simple dietary restrictions, you're not going to get that. So, and even simple fasting cycles, you're not going to get that. So then that's something that we're very interested in.
This is fascinating to me. The idea of the cycling, it seems really important in all of this, because it seems like, I guess, single periods of starvation in a mouse, for example, how long do the beneficial effects last? And therefore, presumably that's why you get into cycling fasting in various ways.
They last pretty long, right? So we published a couple of years ago one paper where we gave mice a high-fat, high-calorie diet, very bad. It become big very quickly. And then just once a month for 4 or 5 days, I forgot, I think 5 days, we give them a fasting mimicking diet, right? And then when we look at the fat, I think 14 days later, but also when we look at the ketone bodies 4 days later, after we return to the high-fat diet, they still keep on burning fat, and or still the fat is still in a different, more mitochondrial biogenesis, more oxidative phosphorylation phase, right? So it seems like they're entering, if anything, a fat catabolic, high metabolic mode, right? So, and they keep it, right? And then of course we reverse using this, we reverse the effect of high fat, high calorie diet on longevity, heart function, cholesterol, glycemia. So it's really remarkable. Now we're actually testing exactly that now in a big trial in here in Southern Italy. That's why I came. 500 people, 3 groups. We already recruited 200. So one group does nothing. And this is Southern Italy. This is like one of the worst areas for obesity and diseases in all Europe. And 167 do a regular diet, 167 do a fasting mimicking diet once every 3 months, and 167 they do both the fasting mimicking diet and the longevity diet, which is this diet that I came up with to sort of put together everything we've ever learned from lots of different labs.
Can you give me a summary of the outcomes for either different diseases or functional aging? Really just a summary of what you've seen so far.
Yes, so I think it's pretty clear with the normal subjects or the relatively normal subjects that we get if they're not taking drugs, we get a reduction in blood pressure. We don't seem to be getting a reduction in blood pressure in people that are mildly hypertensive that are taking drugs, hypertension drugs. We instead clearly see a reduction of A1C, and this is over and over in now a number of clinical trials. A1C in the normal, but particularly in the ones that are pre-diabetic, and now clearly in the diabetic, right? So now there is a new trial from Leiden that is about to come out, and several others. So that's pretty clear. So I would say that now we have hundreds of people for diabetes and pre-diabetes, and I would be shocked if those were not. I don't want to use conclusive, but certainly let's say it's pretty close to conclusive, right? Cancer is, it's gonna take a while, because I think that there's a lot of cancers, a lot of therapies, a lot of modalities, right? So yes, unfortunately, it's just very tough to say anything about cancer. Certainly, it worked well with the first randomized trials with clinical response and pathological response. But then overall 5-year survival, we're not really seeing much of an effect. And I don't know, I mean, of course, these were early stage patients that received surgery. So, you don't necessarily expect a big effect of survival. I mean, you just give them chemo with or without the FMD early on before surgery. So then you're waiting 5 years. Some of them didn't, many of them did not comply to the diet, right? So I would say that we're very, I mean, in mice, it just works extremely well in our lab and in many labs. And no matter what the cancer, no matter what the treatment, which is really hard to believe almost. But in people, I don't know. We just have to wait and see, right? So the cancer is going to take a while to. It looks very promising, but it's going to take a while, and we're just going to have to wait and see. There's, inflammatory bowel disease and Alzheimer. We're about to finish one, and multiple sclerosis. We finished one, but those are early, and I mean, the first trial was fairly positive with a single cycle of fasting mimicking diet, but now there's one that just finished in Genova and another one starting in Los Angeles. So so I would say that we're still early on with those. But I think in general, it's just clearly going after the aging risk factors and markers, and just over and over and over, whether it's IGF-1 or C-reactive protein or blood pressure. And it doesn't, differently from calorie restriction. If you look at the calorie restriction data, a lot of the times you may have somebody starting with, say, glycemia of 85, and they end up at 70, right? You don't see this with the FMD. If they're 85, they're probably going to stay at 85. If they're 103, they're probably going to go down to 85. I mean, not all the times, but I say pretty consistently, right? We see this, at least as an average. As an average, we keep seeing this. And so it just seems to be more going after the fundamental aging changes and just bringing them back to a more youthful state.
Super exciting. Let me ask you about your own sort of personal career progression. Let's go back to the start. Let's go back to the beginning. How did you get into science in the first place, Valter?
I got into science when I was a music student in Texas, and they asked me to direct a marching band as part of the music program at University of North Texas. It's a famous jazz school, and I said, "There's no way I'm doing that." I was a rock guitar player, and they said, "Well, if you don't do that, then you're out." So then in my head was like aging, and so immediately I went to the biochemistry department, and I said, "You know what? I've always been interested in aging." And and the chairman, I think Dr. Norton back then, he was laughing. He's like, "You're never gonna make it in biochemistry." But sure enough, I joined the biochemistry department, and then that's all I've ever done. I was 19, I think.
When you say you were always interested in aging, you mean as a kid, or as a teenager, or what? I mean, why? Why would you be interested in aging as a young person?
I mean, I always assume that it's got to do with somebody dying, probably my grandfather. And 5 years old, I saw him dying. I mean, I actually physically saw him dying. So I think that maybe in the head of a 5-year-old, that's just like, it tells you, there may be something bigger than everything else that you have around, right? Whether you live or die. So probably, I mean, I never thought about it very much until maybe I was 16, 17, 18. And then I'm imagining that was in my head in such a fundamental way that as soon as I had an opportunity, there was it.
Let's fast forward to when now you've got your credentials and you choose to work on not humans but yeast, right?
My first project was Roy Walford, and there was a disaster. The 2 years of comparative biology that didn't go absolutely anywhere. I was very depressed. Because we are comparing young mouse and old mouse and young blood and old blood. And so then after 2 years of suffering like that, at a certain point you realize we need something much simpler than this, to figure this out, right? So then from Walford back to bacteria, and then starving bacteria with Steve Clark, and then starving yeast with John Valentine. I did more in a year working on yeast than I did than I would have done probably in 5 or 6 years working in Walford's lab, even though I only stayed 2 years.
Just explain how you starve a yeast cell. This is a single-celled organism. What does starvation mean there?
Yeah, so for both E. coli and yeast, it means that you grow them as a population and you just let them be, probably not too differently as we then eventually tested from what happens in the rotting figs and rotting grapes from where these yeast were isolated in Northern California, in your area many years ago, right? So the yeast usually get lots of sugar, and then they grow very rapidly, and then they run out, and they wherever they end up, they sit there in that medium, right? So it's pretty straightforward. We just let them be in their fairly natural self-generated environment. So the yeast will utilize this glucose and they generate a lot of ethanol. And then they live off the ethanol and the acetic acid, right? It's like ketone bodies / ethanol environment that's self-generated. And then whether we did it with the lab strains or we did it with the ones from the wild, we got exactly the same effects. Yeah.
You're almost like the poster child, Valter, for someone who has applied the basic science to real human clinical research and interventions in humans, both in cancer and in aging as well. So was there any point where you said, that's it, I'm doing this, we're going into humans? Or, why not just go back to the lab and do the mouse work and do the yeast work and be happy publishing, great papers? Was there something driving that transition?
I always thought eventually maybe I'll go to medical school and I'll do a PhD, MD, but I never did that because, of course, I liked it too much when I was doing science. But yeah, I think that I started with mice and humans, and then the humans. I love science, but I really always liked this applied idea. I mean, I wanted to solve a big problem, right? I didn't want to solve a yeast problem. I want to solve a yeast problem to solve a human problem, right?
So our listeners are probably wondering how they can tap into all of this. You've written a book. I mean, what are the resources that are available to people to make a go of this?
Yeah, so the longevity diet book is really lots of 30 years of my work and lots of people's work in the field, and all the way to the clinical application and the epidemiological studies, and so focus on aging and longevity and applying this to lots of different diseases, prevention and treatment. It talks about both the everyday diet and the fasting mimicking diet and how they're used in combination or one or the other to attempt to help you delay aging and protect against age-related diseases. Then there's a new book coming out. There's focus on all of this, the longevity, the everyday diet, as well as the fasting mimicking diet and cancer. And it talks about 10 clinical trials. And of course, these are best implemented if you contact the Create Cures Foundation Clinic, and that's at createcures.org.
As you look around now at the field, it's really moved so far from when we started. What do you think is the most exciting thing right now in aging biology?
I mean, I really like the epigenetic reprogramming, Belmonte and David Sinclair and others, and so I think that's really exciting. And of course, we didn't talk about it, but the fasting mimicking diet. One of the things that does in the shrinking starvation process is turns on Yamanaka factors, right? So we're very interested in ourselves in tapping into that, and is it can the starvation refeeding cycle activate some of these epigenetic reprogramming mechanisms, and so I really like that. But of course, there's lots of different ideas, and so I think that that's good. We'll see what happens in the next 10 years. Certainly a very exciting moment to think different. Yeah.
Yeah, no, I have to agree with you, Walter. This is really exciting. We actually had David Sinclair on the podcast recently, and his amazing work, as you point out, on sort of reprogramming and recovery of function, and just to hear the work that went into that, postdoc slogging away, trying different factors in different ways has been fascinating. So it's fantastic that you're now connecting with that entire area in the sort of relationship between diet and reprogramming.
Yes, so we actually started a while ago, and I think the first paper was 10 years ago, where we look at the blood system, and we were using chemotherapy and fasting mimicking diet, and we saw the hematopoietic stem cell being turned on and having a clear advantage if the mice were fasted in generating, in self-renewing, so stem cells making more stem cells. And then eventually we started to see what looked like reprogramming in the pancreas. And in another paper, this was 2016, I believe, and we saw that we could damage the pancreas stopped making insulin, and then we will start fasting mimicking diet, refeeding cycle, and we will see the pancreas, the islet cells being reprogrammed into making insulin again, even though they were terminally damaged. Right. So then we started looking at the genes, and some of these genes were in fact Yamanaka factors. Right. So, and they would be turned on temporarily, and then for maybe 1 or 2 days, and then come down. And then we went on to show this in the gut. Now we're about to publish about the kidney. We've showed some of the same effects in the nervous system. Yeah. So we published actually many papers on this FMD fasting dependent turning on of this what seems to be epigenetic reprogramming, or certainly cellular reprogramming, leading to a rejuvenated, more functional state. Yeah.
This nexus between aging interventions which slow down aging processes but also stimulate regeneration is absolutely amazing. It's absolutely, I don't think we would have predicted this a few years ago.
No, probably not.
One final question is, what did you have for breakfast this morning?
Breakfast, I mean, I'm in southern Italy. I normally have tea and this almond spread, and then a frisella. That's my all year round. Now here I don't have it because I'm not at home. So I had this basically milk and a frisella, which is this whole grain bread from southern Italy, and I highly recommend it.
Fantastic. Thank you again, Valter, for your time. It's been really delightful to talk to you.
Thanks, Gordon. Great to talk to you.
Thank you for listening. Please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts. We're Not Getting Any Younger Yet is produced by Vital Mind Media. The Buck Institute's very own Robin Snyder is the executive producer. Wellington Bowler is right next to me here, directing the recordings. Stella B is behind the scenes, ready to debrief when we wrap. And the esteemed Sharif Izzat weaves the show together for you. If you're listening to the podcast, you'll know that there has never been a more exciting time in the research on aging. Discoveries in the lab are moving to the clinic to help us all live better, longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to learn more and to donate.